But we've got a problem. We have a lot of drugs that are pretty good at killing the bulk of tumor, but we don't have drugs that specifically kill the stem cells that are driving that tumor. So what I'm going to tell you about is some work out of my laboratory that we got interested in this problem about 6 years ago, that in order to prevent metastasis, in order to prevent recurrence, we really have to eliminate these stem cells that are the drug-resistant source of this continuing problem. So we turned to a receptor that is interestingly found on many stem cells in our body, and that is a member of the LGR4/5/6 set of receptors that were discovered originally by Hans Cleaver's group in the Netherlands, and have turned out to be extremely important because they mark active stem cells in essentially all of our rapidly dividing epithelial systems and in some of our slow dividing epithelial systems. So RSPO1s are the natural ligand for this set of receptors, and the RSPO, there are 4 of these RSPOs, and what they do is they simultaneously bind to one or another of LGR4/5/6, in this case I've shown LGR5, and a co-receptor. That co-receptor's job is to continuously degrade frizzled receptors which control Wnt signaling that drive stem cells. When a ligand binds, it shuts off the ubiquitin ligase activity, and you get much higher levels of frizzled receptors. Well, that's all nice, but that doesn't help us target stem cells. So what we did is focus in on just the LGR5 receptor itself. And this is a receptor which is not particularly widely expressed in prostate cancer cell lines in the DepMap collection of 100 or 1,000 or so cell lines. But LGR4 is highly expressed in prostate cancer, and a few prostate cancer cell lines express LGR6. So we're developing then a drug which will bind to LGR4 in prostate cancer cells and kill them. And what this is a drug now called Cymirafin. It consists of an immunoglobulin domain at the N-terminal end, followed by the receptor binding domains of RSPO1, and then a system for linking a toxin to the end of the molecule. We use an enzyme called sortase, which very specifically puts this toxin at the end, and you wind up with exactly 2 warheads on this molecule. So now we ask, are we getting true selective killing of these stem cells compared to all other cells? So we engineered a couple of standard cell lines to overexpress, in this case, LGR5, and you can see that we have about 1.5 logs of differential cytotoxicity, and that's also true for another pair, in this case, ovarian cancer cell lines that we engineered to overexpress those LGR5. But it also kills true stem cells. So we can detect the presence of stem cells using spheroid assays, because only a stem cell will form these kinds of assays. And we have very, very good potency against these stem cells in multiple different solid tumor models. The pharmacokinetics of this molecule look attractive. It's got a reasonably long terminal half-life, long enough to support a weekly dosing regimen. And if we take a tumor that has low levels of these receptors and we grow it in a mouse and don't treat it, we get the black curve. If we treat it, we get some activity due to the presence of low levels of of these receptors in this particular cell line. But when we engineer that cell line to overexpress one of these receptors, LGR5, it grows a little faster, but we also get a much, much larger therapeutic effect. There's a nice dose-response effect in xenograft models with even very, very low doses producing 40% or so reduction in the tumor growth rate. So here's an example of one of the models that we're testing. This is a very aggressive gastric carcinoma. And in this case, when we give 4 weekly doses, we have most of the tumors growing out, but very few and essentially none of the treated animals grew out a tumor. We see this in another model, which also has high levels of Wnt signaling, in this case, a neuroblastoma, where this is selected because of this extraordinarily explosive growth of this tumor, and yet a couple of doses of the drug delay the onset of this explosive growth very highly. One of the virtues of being able to use an enzyme to load a warhead specifically on a drug, on the molecule, is that we can load multiple different kinds of warheads. We use MMAE in most of our models, but we can load a anthracycline on it. We can load deruxtecan, famous because of its success in trastuzumab and breast cancer. And we are exploring whether we can then have disease-specific drugs to disease-specific warheads that we can use more widely. So where it stands now, it does kill stem cells. There are no other stem cell-specific drugs that have ever been approved. It's a novel concept of using a natural ligand, and we did that because everybody else's attempt to develop antibodies to this set of receptors has not worked, and none of those have entered the clinic at all. It's very high affinity. It's got selectivity, both in vitro and in vivo. It kills real stem cells and has very little toxicity. I haven't shown you the toxicity data, but it's got a very, very wide therapeutic index. We're not getting much toxicity at doses that are clearly effective in the animal models. It's got favorable pharmacokinetics. Exploration of how to produce this in a commercial CHO clone is underway. We've produced research clones at this point, so we can definitely make the drug in a commercial type setting, but we're going to have to look to companies that would want to license this drug to do a commercial production. It's got a very limited workflow in terms of the purification and production of the molecule. And because this is an ADC-like molecule, we have a pretty well-paved regulatory route through the FDA. So this project is at the point where it really needs to be taken out of an academic environment and out-licensed to a company that has enough funds to take the next stages in drug development. And I hope this is a molecule that eventually you all will hear more about as we begin to address this opportunity in prostate cancer because of its high level expression of LGR4. Thanks so much.
Thank you, Steve. Wow, stem cells have been the nemesis of everything we've been dealing with for a long time, at least in a lot of cancers and prostate. We talk about the whole concept of intermittent therapy was that you had sensitive cells, you had sort of moderately sensitive, and you had the insensitive stem cells, and you let those let those grow up, and then you got a problem. As Steve said, there's been a lot of focus on trying to stabilize stem cells, get rid of them, inactivate them, whatever you want to do with them. The only positive thing I've heard in prostate cancer, quite honestly, in the last 10 years have been some of the work that was done with radium-223, and it did have some effect on stem cells, but not a lot. So I think we may have heard this all works, a Nobel Prize speaking thing by Steve here, so congratulations on that. We will have you back next year if Dr. Gladey that keeps us informed on how this is going.